Can biomolecules last that long? Last week, we noted that there was increasing evidence, first from Mary Schweitzer and now from others. In the background of this slide is one of the others, that there are original biomolecules still left in dinosaur bones. We also noted that there was widespread disbelief that molecules could last for 65 plus million years. The question that we raised today is there any evidence behind that disbelief, or was it always merely superstition? And before we go on, we should probably ask what biomolecules are we talking about? Well, there's an article by Mary Schweitzer et al. In 2013 called Molecular Analysis of Dinosaur Osteocytes Support the Presence of Endogenous Molecules. And this is one of the evidences for those biomolecules, and you're going to be, I think, interested in seeing the evidence. And in this case, it will be actually seeing the evidence. The discovery of soft, transparent microstructures in dinosaur bone consistent in morphology with osteocytes was controversial. We hypothesized that, if original, these microstructures will have molecular features in common with extant osteocytes. We present immunological and mass spectrometry evidence for preservation of proteins comprising extant osteocytes, specifically actin, tubulin, PHEX, which we'll get into later, and histone H4, in osteocytes recovered from two non-avian dinosaurs. Furthermore, antibodies to DNA show localized binding to these microstructures, which also react positively with DNA intercalating stains, propidium iodine and 4,6-diamidino, 2-phenylenediol dihydrochloride. Each antibody binds dinosaur cells in patterns similar to extant cells. These data are the first to support preservation of multiple proteins and to present multiple lines of evidence for material consistent with DNA in dinosaurs, supporting the hypothesis that these structures were part of the once living animals. We propose mechanisms for preservation of cells and component molecules and discuss implications for dinosaurian cellular biology. And what you see there is are several osteocyte-like structures. If you're familiar with osteocytes, these ones in particular look very much like them. But all of them look somewhat like them. The top three are from Tyrannosaurus rex, and the bottom three are from Brachiolephosaurus canadensis.
Now that's bone tissue?
That is bone fossils that have been demineralized, and what they found inside of it were these structures. Now, the structures can also be looked at with scanning electron microscopes, and you can see that there's cells with a lot of little filaments sticking out of them. And in some cases, particularly in this case, you can see there's the pods that are sticking out of them are actually interwoven with the rest of the bone, specifically with the collagen. And they showed transmission electron microscope views. And here you can see that the innards of these things, particularly the B. canadensis, have been largely evacuated, but you can still see the outlines of the cell membrane with some materials kind of stuck in next to it. And just for comparison, here's some ostrich bone with osteocytes inside of them, and you can see that there are openings between the osteocyte and the matrix around it. So that part of it is not that terribly surprising. Now, here's what's more interesting, and that is they have taken antibodies to actin and put them on Tyrannosaurus rex and then put fluorescent antibody, and you'll notice that the antibodies to actin have tended to concentrate in the cell. Now, any old antibody doesn't do that. And so in B, you can see no fluorescence. This is, again, another Tyrannosaurus rex cell. And here's what ostrich looks like under the same treatment. So the fluorescence is expected. This is to actin, and then the bottom ones are to tubulin, the same setup. Tyrannosaurus rex with the antibodies, a control, and an ostrich control. Where you can see the ostriches light up quite nicely. The Tyrannosaurus lights up quite nicely. The Tyrannosaurus won't light up with just any old antibody. Well, you have a question in the back.
Would actin be present in bacteria?
Some bacteria have an actin-like protein, and so this is not necessarily specific for osteocytes, but we're going to get to some where it is specific. Here again is Brachiosaurus canadensis, and again you can see the antibodies to actin light up. The ostrich lights up. Any old antibodies don't light up. And again, to tubulin, the same thing. Now, I may be wrong, but if I remember correctly, tubulin is not found in bacteria. And phosphoendopeptidase is definitely not found in bacteria. That is an enzyme that helps form bone. And if you'll notice here, the T-rex and the B. canadensis both light up. These are the same cells, of course, just one in dark field and one in standard transmission microscopy. And interestingly enough, when they took a piece of bone from B. canadensis and put antibodies into it, and then the antibody, the osteocyte inside lit up.
Which means?
Well, it's probably a real osteocyte. It has the things that are necessary for osteocytes, and this particular enzyme is found only in, and in fact this is a monoclonal antibody, so it's found only in birds today. And in fact, here you can see ostrich lighting up, and interestingly, alligator does not. That's an alligator osteocyte, and you can see it does not have the green fluorescence. Now, they went ahead and did some more stuff, and this is anti-DNA antibodies that can detect up to 4 intact segments of DNA. And you can see T-rex has some in what looks like a nucleus. B. canadensis also has some, and ostrich has some. Ostrich has more, of course, which is not surprising given that it's relatively new material. Again, they stained it with propydium iodide, which is known to stain DNA. And you again get staining in all 3. And finally, you get 4,6-diamidino 2-phenylenediol dihydrochloride. And again, all 3 of them will stain very nicely. That suggests that there is DNA in those dinosaur osteocytes. Now, if you read their article, there's a note added in proof. A recent paper by Ellentoft et al., 2012, hypothesizes a half-life of DNA of 520 years, more or less, in an optimal depositional environment, suggesting that DNA should be degraded to single bases by a little more under 7 million years, even though they also state that considerable sample-to-sample variance in DNA preservation could not be accounted for by geologic age. What they are noting is that this should all be gone by 7 million years. At least according to the paper. Their half-life estimate was based on extrapolation of data taken from over 150 relatively recent Holocene bones, less than 10,000 years old. Fossils older than this were not examined for DNA. Well, partly because they didn't have any fossil bones older than that. All of our assays require at least 4 or more bases to generate reactivity, particularly within the intercalating stains. While ultimately sequence data is required to verify the endogeneity of this material, it is unlikely that 4 independent assays, each capitalizing on different aspects of the chemistry of DNA, would show identical patterns of localizations interior to these cellular structures and different from antibodies to various proteins. Therefore, we suggest more rigorous testing of extrapolation models on actual fossil material from older specimens. Additionally, we have found that chemical reactions interfere with analysis of DNA and proteins and should be taken into account before assigning a definitive half-life. Yes, Ariel.
4 bases. I'm not following that. You get 4 bases anywhere. I mean,
plus the phosphate sugar backbone to tie them together.
okay.
In other words, this is DNA that's intact at least to 4 base length. And you see this should be out at single base by the time you get to 7 million years. Now notice what she is doing. She is saying you can't trust the material that we have a definitive time for. That extrapolation is not a fair one. And so what we need to do is take geologic samples. Think about that and what the likely answers would be if you did that. Well, what about that? Oh, I've got an M in front of that. It should be Ellentoft, M-E. That article. Well, let's go and look at it. It's available on the net as well. And the abstract is, claims of extreme survival of DNA have emphasized the need for reliable models of DNA degradation through time. By analyzing mitochondrial DNA from 158 radiocarbon-dated bones of the extinct New Zealand moa, we confirm empirically a long hypothesized exponential decay relationship. The average DNA half-life within this geographically constrained fossil assemblage was estimated to be 521 years for a 242 base pair mitochondrial DNA sequence, corresponding to a per nucleotide fragmentation rate of 5.50 times 10 to the minus 6 per year. With an effective burial temperature of 13.18 degrees centigrade, the rate is almost 400 times slower than predicted from published kinetic data of in vitro DNA depurination at pH 5. In other words, it's slower than they thought it would be when they tested it in the laboratory, probably because the pH is different and because bone has a protective function in this case. Although best described by an exponential model, considerable sample-to-sample variance in DNA preservation could not be accounted for by geologic age. And you're going to see some of that variance in just a minute. This variation likely derives from differences in taphonomy and bone diagenesis, which have confounded previous less spatially constrained attempts to study DNA decay kinetics. Lastly, by calculating DNA fragmentation rates on Illumina high sequence data, we show that nuclear DNA has degraded at least twice as fast as mitochondrial DNA. So nuclear DNA is worse than mitochondrial DNA in this particular setting. These results provide a baseline for predicting long-term DNA survival in bone. And there's the data. You can actually download that slide if you want. And you'll notice that, yeah, you have a correlation. Now, the R is not very great, but it's there. And out to this point here, I would say that the carbon-14 dates that they're using to give the age are probably within 10%. Out to about here, the carbon-14 dates are probably within 1%. This is where the Nineveh Bones data falls, just about in this range here. Actually, I think just before this. But it's only off by 150 years if the data that we have is correct. So that this is all pretty good. If you were to shift it over 150 years, it wouldn't make much difference. And you can see that all three sites have a slope, and the slopes are not statistically different from each other, although they do look slightly different if you're measuring them. And here is the old estimate, and you can see that the new estimate is considerably slower in terms of degradation than the old one. Or another graph that they have will show you the half-life is 521 years here. You're curious as to why Alan Toft was doing all this stuff. Well, it turns out that Alan Toft is interested in a particular problem that you have, and that is that New Zealand has moas, or had moas, before the Maoris destroyed them. And the moas didn't have wings. So how'd they get to New Zealand? And some jokester called it Noah's Ark. And some people thought that they were volant, that is to say they flew there and then lost their wings. So how exactly did they get there? And that, of course, is a problem for creationists as well, because the moas certainly didn't walk across the water to get to New Zealand. And it's a problem because you have moas in New Zealand, you have cassowaries in northern Australia and Papua New Guinea, you have emus, and you have rheas in South America. And if you try to make them all descended from one, well, how do you get there? So there are a lot of unanswered questions, and he's interested in what kind of DNA did the moas have so that he can make some kind of estimates as to what other... there are also kiwis in New Zealand. And are they related to the kiwi? Are the kiwis and the moas related to each other, or are they two separate sets of birds? Well, it turns out, at least what I can read of the article, that they're two separate birds. But that's why he's interested in getting all that DNA from those moas. He's trying to figure out how they're related to each other. Anyway, that one is also on the net, although it's a pre-publication thing. Interestingly, Nature News has an article called DNA has a 520-year half-life, 521 year, which of course is taken off from this, and as you will see, genetic material can't be recovered from dinosaurs, but it lasts longer than thought. So these are people who are arguing that the dinosaur data has got to be wrong somehow. And that one also is available on the web. Few researchers have given credence to claims that samples of dinosaur DNA have survived to the present day. But no one knew just how long it would take for genetic material to fall apart. Now, a study of fossils found in New Zealand is laying the matter to rest and putting an end to hopes of cloning a Tyrannosaurus rex. So what Mary Schweitzer got, it just simply can't be gotten. Skipping through the article a little bit, it's a relatively short article, but paleogeneticists led by Morten Allentoft at the University of Copenhagen and Michael Bunce at Murdoch University in Perth, Australia. I would have thought that Allentoft lived in New Zealand, but apparently not. Examined 158 DNA-containing leg bones belonging to 3 species of extinct giant birds called moa. The birds, which were between 600 and 8,000 years old, had been recovered from 3 sites within 5 kilometers of each other, with nearly identical preservation conditions, including a temperature of 13.1 C. The findings are published, as we said before. By comparing the specimens' ages and degrees of DNA degradation, the researchers calculated that DNA has a half-life of 521 years, and then 3/4 or 1/4 life of 1,042 years and so forth. Pardon me, 1,042 years. The team predicts that even in a bone at ideal preservation temperature of -5 degrees centigrade, effectively every bond would be destroyed after a maximum of 6.8 million years. In other words, it's not going to last that long, especially at 20 or 30 degrees centigrade. This confirms the widely held suspicion that claims of DNA from dinosaurs and ancient insects trapped in amber are incorrect. Pay no attention to those data that are supposed to have been gotten out of insects. They just can't be right. They just can't. Said Simon Ho, a computational evolutionary biologist at the University of Sydney in Australia. However, although 6.8 million years is nowhere near the age of dinosaur bone, which would be at least 65 million years old, we might be able to break the record for the oldest authentic DNA sequence, which currently stands at about 500,000 years. Now, Schweitzer has put it at 70 million years or something like that. But notice that claims of DNA from dinosaurs and ancient insects trapped in amber, there's a bunch more that's like 30, 40, 50 million years old. So Schweitzer's not alone in her finding of DNA. The calculations in the latest study were quite straightforward, but many questions remain. I'm very interested to see if these findings can be reproduced in very different environments, such as permafrost and caves, said Michael Knapp, a paleogeneticist at the University of Otago in Dunedin, New Zealand. This guy does live in New Zealand. Skipping, I think that's the last paragraph. Yeah. And there is, in fact, cited by one of the articles, an article by Willerslev et al. From deep-sea ice cores. Apparently they drilled all the way down, and they found plant material and insects, and were able to get DNA out of them. So we're getting DNA, but we're not supposed to be able to get DNA. Then there's an article by Christine Nielsen March, Biomolecules in Fossil Remains, Multidisciplinary Approach to Endurance. And this one in the middle of it, Christine March is from the University of Newcastle upon Tyne. Identifications of the mechanism and controlling factors for biomolecular preservation in ancient material are the main objectives of the Ancient Biomolecules group at the University of Newcastle upon Tyne. The ABG has an interdisciplinary and systematic approach to biomolecular preservation and works in close collaboration with colleagues studying ancient DNA, Dr. Alan Cooper at the University of Oxford, ancient proteins, Dr. Peggy Ostrom, Michigan State University, and the application of synchrotron radiation to biological materials. So these are people who their business is studying how long these biomolecules will last. And in that article, there's a fascinating table. And this is the half-life for DNA, collagen, and osteocalcin. And you can see that the detection limit is supposed to be 125,000 years. 17.5 thousand years, and at 20 degrees centigrade, 2.5 thousand years. Now, interestingly enough, if it's 10 degrees centigrade and it's 17.5 thousand years, and there was a flood, let's say 5,000 years ago, more or less, that's within range. So, if you had a flood and relatively cool conditions afterwards, there's no reason not to be able to find DNA. As you can see, collagen lasts quite a bit longer. That's 2.7 million years for very cold material, 180,000 years. That should easily be, yes?
Yeah, as to what you were saying, I've heard some estimates that the original ice sheet came all the way down as far as Kansas City.
Yes.
So it would have been extremely cold. And that's, what, 4,000 miles from the Great Circle, or more than that.
Yes.
And all that shrunk back before the modern age, so it's been very cold and this stuff might have been preserved.
That's right.
Suggesting.
Now, one thing to keep in mind, though, is that if you're modeling the standard model, the Ice Age started some 4 million years ago. And so if you're talking about Eocene material, it has to survive from, say, 50 million years to 4 million years, which means it has to survive 46 million years under conditions that are usually thought to be pretty warm, because the plants and animals that form the assemblage usually live in warm climates. And if you're talking about dinosaurs, you had to survive in climates that were usually assumed to be fairly hot.
Assumption, feelings, and I think so, not hypothesis, and not really scientific.
Well, the point that I'm making, yeah, the point that I'm making is that this is one case where evolutionary predictions are clearly in opposition to a creationist, a short-age creationist anyway, prediction. And so, you know, because short-age creationist predictions would not have any problem with having collagen left. Osteocalcin, on the other hand, might actually last 110 million years if it's kept cool. But of course, usually one would assume that during the age of the dinosaurs, 20 degrees centigrade would be more like the temperature you'd see, and so there, 1 million years and it should be gone.
One other thing, I haven't looked at it for several years, so it's not right off the tip of my tongue, but one very prominent scientist, which we all know had quite an important part in the discovery of DNA and so forth, said that you have to put carbon-14 into the particular chemicals or cells that you find, and that half of these DNA chemicals do not have carbon-14 in them. So it's not part of the makeup for the survival of that cell. It's gotten in there outside of the actual reproduction or the creation of the creature itself. So they're going to find a lot of, according to that, they would find a lot of these cells with these chemicals. It doesn't have carbon-14 in it, which means that they just decay, but not radiometrically. They just rot away, or you had a term there, it's not coming up just real quick in my head.
Well, they hydrolyze, and that's
that's probably
And that's a non-radiometrically
Yeah.
determined
They normally
process.
just disappear with time.
Right now, approximately 1 in a million, 1 in a trillion atoms are carbon-14 of carbon. And so after all of that's decayed away, that would only affect 1/1,000,000th of your DNA. So it's probably not going to be a significant factor. This is not a radiometrically determined process. It's more of a hydrological, and that's why it varies so much with the temperatures, because the hotter the temperature, the easier it is for water to break up the bonds, excuse me. Anyway, well, of course, if that were left unchallenged, then you'd have to concede that what Mary Schweitzer found just isn't really there. And so Schweitzer and her colleagues came back with the idea that iron and oxygen chemistry might preserve soft tissues from deep time. And that one is also available on the web, if you're interested. The persistence of original soft tissues in Mesozoic fossil bone is not explained by current chemical degradation models. Notice the frank admission that what we're finding shouldn't be there. We identified iron particles, goethite alpha FeOOH, associated with soft tissues recovered from 2 Mesozoic dinosaurs using transmission electron microscopy, electron energy loss spectroscopy, micro X-ray diffraction, and iron micro X-ray absorption near edge structure. Iron chelators increased fossil tissue immunoreactivity to multiple antibodies dramatically, suggesting a role for iron in both preserving and masking proteins in fossil tissue. You chelate out the iron and it's easier to detect the tissue. Hemoglobin increased tissue stability more than 200-fold, from approximately 3 days to more than 2 years at room temperature. In an ostrich blood vessel model, developed to test post-mortem tissue fixation by cross-linking or peroxidation. Hemoglobin-induced solution hypoxia coupled with iron chelation enhances preservation as follows. That is, if you have hemoglobin plus oxygen, you actually get better preservation than hemoglobin without oxygen, which is better than no oxygen at all, which is much better than having oxygen but no hemoglobin. The well-known oxygen-heme interactions in the chemistry of life, such as respirations and bioenergetic, are complemented by oxygen-heme interactions in the preservation of fossil soft tissues. Now, transmission electron microscopy shows iron intimately associated with vessels recovered from demineralized dinosaur tissues. I'm just going to read a few kind of random quotes, well, not random, but scattered quotes, to give you some idea of what the article is about. We discussed this article some last week as well. Synchrotron microprobe techniques were used to investigate dinosaur and ostrich vessels, intravascular material, and chemical speciation of associated iron. So they're using some fairly sophisticated techniques to find out what's going on. Ostrich vessels were incubated in a concentrated solution of red blood cell lysate to approximate post-mortem erythrocyte lysis. I'd like to know exactly how concentrated, but control tissues were prepared identically and then incubated in either sterile distilled water or phosphate buffered saline solution. Hemoglobin-treated vessels have remained intact for more than 2 years at room temperature with virtually no change, while control tissues were significantly degraded within 3 days. Of course, 2 years is a far cry from 67 million years, but and then there's a comment at a blog site for which I'm indebted to Ariel Roth and through him Sean Pittman, an explanation that is not exactly ironclad. It's written by Jay Weil, a PhD, who got his PhD in nuclear chemistry at the University of Rochester. And his comment is in 2005, Dr. Mary Schweitzer shocked the scientific world by reporting soft tissue in a Tyrannosaurus rex fossil that is supposed to be 68 million years old. While many scientists who are more interested in their preconceptions than they are in the data tried to dismiss her findings, yes, they're really there. Several other examples of soft tissue in fossil that are thought to be millions of years old have been found, and he has links to several other reports. In the end, it has become nearly impossible for a thoughtful scientist to conclude anything other than the fact that there is soft tissue present in some fossils which are thought to be millions of years old. Now, for someone who truly believes in an ancient Earth, it's very hard to explain how soft tissue can remain in a fossil that has been in the ground for millions of years. Even for a young Earth creationist like myself, it is still a difficult thing to understand, but not quite as difficult. Soft tissue tends to decay in a matter of days or weeks. From a chemical point of view, it is hard to understand how it can stay soft for even a few years, much less hundreds, thousands, or even millions of years. Fortunately, Dr. Schweitzer has continued her studies on soft tissue in dinosaur fossils, and she has found at least one chemical mechanism by which soft tissue can be preserved for significantly longer than anyone expected. And that's, of course, the article that we just looked at. She and her colleagues began by examining a soft tissue from her T-Rex fossil as well as a Brachiosaurus canadensis fossil. I wish I was a kid. I could pronounce that easily. While the T-Rex fossil is supposed to be about 68 million years old, the B. canadensis is supposed to be about 76 million years old. Nevertheless, under a transmission electron microscope, both are seen to harbor soft vessels that are probably blood vessels. Interestingly enough, however, the vessels have tiny particles of iron embedded in them. Where did the iron come from? It came from the dinosaur's blood. When the dinosaurs were alive, the iron was tightly bound to a blood protein known as hemoglobin. Once death occurred, however, the hemoglobin decayed, releasing the iron and allowing it to mix with the tissues. How does this help with preservation? Well, most biological molecules are polymers, which means they're long chains of repeating or almost repeating chemical units. Proteins, for example, are long chains of smaller chemicals called amino acids. It is well known that iron, especially when it has the chance to react with oxygen, can cause polymers to link together in a process called cross-linking. When polymers link together like that, they are more resistant to decay. Since Schweitzer and her colleagues saw iron particles embedded in the soft tissues of the dinosaur specimens, they thought that perhaps the iron was cross-linking the biomolecules, and that's at least part of how the soft tissue is being preserved. They decided to test this hypothesis by taking the ostrich blood vessels and soaking them in one of three liquids: concentrated solution of blood, whose red blood cells have been destroyed, basically freeing the hemoglobin, a pH-controlled saltwater solution, or sterile distilled water. They found that while the blood vessels soaked in distilled water and salt water degraded significantly in a matter of 3 days, the ones that had been soaked in the blood concentrate were intact after sitting at room temperature for over 2 years. Obviously, then, something from the blood was able to preserve the vessels. As Schweitzer and her colleagues explained how soft tissue can be preserved in fossils that are millions of years old, if you read popular sources, you'll certainly get that impression. For example, here are 3 headlines for articles that discuss their study. Mysteriously intact T-Rex tissue finally explained. Iron preserves ancient dinosaur soft tissue in fossils. Controversial T-Rex soft tissue discovery finally has an explanation. And of course, links to the articles that use those headlines. Of course, if you understand the study, you recognize that the press has completely mangled the science, or at least substantially. While the study represents an excellent first step in understanding how soft tissues can be found in fossils, it doesn't solve the mystery of how it could be preserved for millions of years. There are at least 3 things that indicate lots more research has to be done on the issue. Number 1, the ostrich blood vessels were preserved for only 2 years. While this is more than 240 times longer than what happens under normal conditions, it is still a far cry from millions of years. That's his emphasis. Hopefully the authors are continuing the experiment to see how well the vessels remain preserved after a much longer time span. The ostrich blood vessels were stored at room temperature throughout the course of the experiment. While this is a good first step, in order to really see how well this preservation holds, the authors need to vary the temperature in a way that is realistic. I would think that simulating the freeze-thaw cycle that happens every year would deteriorate the blood vessels over a long time span. And that's certainly something that should be done. It's not clear that iron is always associated with soft tissue in dinosaur bones. For example, consider one of the most striking examples of soft tissue preservation in a dinosaur fossil. If you look at the photos in that paper, and there's a link to it, you see no evidence of iron particles. I contacted the lead author of the paper, and he informed me that he saw no iron particles or crystals in association with the soft bone cells in his samples. The only place he saw iron was inside partially degraded tissues. So while iron might help preserve some soft tissue, it's probably not responsible for all soft tissue preservation. In my mind, here's what Schweitzer's excellent study demonstrates. In at least some cases, iron can preserve soft tissues for a significant length of time. How long? That's hard to say, especially since real-world conditions weren't used in the experiment. Nevertheless, the study at least provides us with a starting point for explaining the preservation of soft tissue in fossils. If the study is extended and starts to use real-world conditions, it might explain how soft tissue can be preserved for hundreds or perhaps even thousands of years. I'm not sure how it could be used to explain the preservation of soft tissue over millions of years, but I remain open to that possibility. Now, my own view on all this is last week we noted an article which showed that structures compatible with dinosaur tissue components can be found by others than just Mary Schweitzer. It's being found all over. The argument that DNA and protein can't last millions of years has some experimental data to back it up. Maybe not ironclad, to use a phrase, but at least some data. It seems to me that the preservation mechanism proposed for 65 million years doesn't look really adequate, at least not to me. I think both sides of this argument appear to be right. And if they're both right, it raises the question of whether the time conventionally assigned to the dinosaurs is correct. Maybe they were alive much later than usually thought. And that would explain why they still have soft tissue in them. But that's my opinion. Now it's your turn.
I got a question.
Just a minute. Let's. Can we get a mic down here? Yeah.
Don't cosmic rays also degrade soft tissue?
As a matter of fact, last week we read a quote by Dr. Jeffrey Bada, who pointed out that not only cosmic rays, but also uranium and thorium, which tend to collect in bones that have been left out for a long time. The dinosaur bones typically have more uranium than you'd expect in them, and more thorium. That radiation also is damaging. And so he claimed that it couldn't last that long because of the radiation problem.
So this recent important scientific video that just came out, Jurassic Park World, shows that the dinosaurs, they're cold-blooded, so they're living in this warm environment, right?
Uh-huh.
So somehow they suddenly got preserved for 65 million years in a very cold temperature that's isolated, that has lots of iron, and is isolated from cosmic rays. Is that right?
Well, it would have to be buried in order to do that,
right.
Yes.
Right.
Yeah, well, of course, the thing that I find interesting is that here's another place where a creationist model has a huge difference with an evolutionary model. In a creationist model, after the flood, the Ice Age presumably followed within probably less than 300 years. Some people would say 0. I don't know if that's really true or not, but I would say certainly a short time frame. 300 years is nothing to preserve DNA. We see 4,000-year-old mummies that have DNA in them. And so it would be interesting to go either to northern Canada or Alaska and look for Cretaceous material, or perhaps Siberia, or maybe Antarctica, and see if the preservation is better there Montana is probably the best place in the United States because it gets the coldest there, but it also gets pretty warm in the summer. So I'm not sure Montana is the best place to look. Of course, Jurassic Park, apparently Michael Crichton knew about Mary Schweitzer's work because, as we noted last week, it actually has been around since 1995 or so. I don't know, does anybody remember when Jurassic Park came out as the book?
He means the original.
The original one. I think it's about...
It's been about 10 years, hasn't it?
If it's been 10 years, then that's 2005, so that would be easily in time for somebody to hear about Schweitzer's work. And of course, even though it was early, you know, you let your imagination run with the possibilities, and then yes, 1997. So that was just 2 years after her PhD, which was done in looking at preservation of blood cells. So yeah, they're thinking about those kinds of things.
I'm not exactly sure all the scientific work has been done on this, but it seems to me, is there a possibility that almost all the carbon-14 was created at the time of the flood? Here we have these masses we can hardly even comprehend of animal life and plant life being destroyed and buried. And maybe there was a lot more carbon-14 at that period when this happened. So there has to be origination for it. So what is that? I'm just suggesting an idea because this is not my field, of course.
Well, the thing to keep in mind is that carbon-14 is not central to this particular discussion. There is also the evidence.
That's what I would think.
There is also the evidence for carbon-14 in these dinosaur bones, and that, of course, I've talked about here in the missing presentation. Doug, you had...
It was actually 1990 is when the hard prover, and then later was republished in 1997.
So 1990. So he was actually ahead of Schweitzer's work, although he took Horner and ran with it. Then he sort of pulled the Jules Verne on us in that case. So Michael Crichton is a prophet before his time. And of course, you know, Mary Schweitzer is constantly having to fight off people who say, well, is there DNA? Well, can we sequence the DNA? Well, you know, can we recreate dinosaurs? And she's, no, there's a long ways to go before we do that. But from a creationist standpoint, you know, if we find a dinosaur in Antarctica, it's probably been frozen since shortly after the flood. The DNA should be in excellent preservation, and we should be able to get something out of it. Ariel.
I take it from that one study that was not published, but at least it, well, it wasn't based. It was based on information from other folks. That last one you showed there, that
In the slide with the table on it. Yeah, that was.
DNA is a challenge to Schweitzer, but the other tissues aren't so much. If you have those cold conditions.
If you have the cold conditions. But see, remember that the hypothesis is Mesozoic was much warmer because it has all these warm climate plants in it and all these warm climate animals. And so the interesting thing is that that people are forgetting that it's not what happened during the Ice Age that's important, it's what happened before the Ice Age if you're doing an evolutionary modeling.
And there is significant data out there that shows the Earth was warmer in the past. at least in the temperate regions, and especially in the extreme poles, than it is at present. But it seems to me that DNA remains a severe challenge to the age of those dinosaurs.
Yeah, I don't know that I can say that it's an absolute slam dunk. You can't account for this, but it's certainly the weight of the evidence seems to be in favor of a shorter age.
In reference to Schweitzer's work, the recent work for adding hemoglobin and iron and so on, keep in mind that iron is the fifth most common element in the crust of the earth. And but in a ways, creationists ought to be happy about something that makes those blood vessels last a little longer than 3 days.
Yes. We needed the preservation too. We just
exactly.
Don't need it as
we
long
we
as
need
they
the
do.
preservation to at least several thousand years, but not to 65 million. And the temperature seems to be against preservation, 65 million.
Yeah.
So
This is a joke. He says,
Just a minute.
You say, well, I wouldn't quite call it that.
Go ahead.
But it seems to me like it's a slam dunk in one way of looking at it, and that you can't duck the evidence.
Yes, Doug.
It's amazing how much smarter I am when I have Google in front of me. It turns out Crichton started writing the book in 1981, but he wrote it as a children's story, and nobody would like it until he eventually rewrote it as an adult story, and so it took him quite a long time to finally get to publishing it.
It's pretty amazing, though. I mean, all he had was Jack Horner's work to model his story on, and somehow he had figured out that there might be. Well, of course, his story was that it was they were getting it from mosquitoes that had been trapped in amber. Which is a little different than going out and hunting the bones themselves.
Where does Schweitzer get her bones?
Schweitzer got her bones from Montana primarily. She worked under Jack Horner for some time. I think he was her PhD advisor. So Horner and Schweitzer, pretty closely linked there. In fact, Horner is the one that told her, well, if you don't think they're red blood cells, then find out what they are. Prove they're not red blood cells. Which is what she set out to. Basically, she set out to try to, well, do they work with this with red blood cells? Do they do that with red blood cells? Do they do? And it's very interesting, as one of the articles pointed out, the stuff that she was doing is totally uncontroversial until you apply it to dinosaurs. I mean, this is how you detect DNA. This is how you detect protein. And how do you detect specific proteins? You use antibodies that are tagged. That's the way you do it. And obviously, it worked pretty well. I mean, the stuff's there. Now, how did it manage to last for 65 million years? Well, the iron story is the best story we've got, I'm not sure that it's a complete story yet. But it's a lot easier if you only have to preserve it for, say, 5,000 years instead of 65 million.
Sure, it looks like a miracle.
Well, one of the things that I thought was interesting is that Mary Schweitzer's answer was, well, test it on known age material that's older than 8,000 years. Well, of course, that's assuming that we know the age and that it's less than, or that it's more than 8,000 years. And if creationists are right, there is no material like that.
What about this kinetic data that Schweitzer refers to in her paper very casually, and that others claim there's no way those molecules could last as long as some of that data you showed here last?
Yeah?
Doesn't that kind of, isn't that data probably more reliable than that study you showed there?
But this kind of argument sounds familiar, doesn't it? Well, we don't know how life evolved, but we're here. I mean, isn't that in essence the kind of argument that we're getting? We don't know how dinosaur tissue can last that long, but it's there. And it's that old, so it's got to last that long. We just don't understand how.
Yeah, but the kinetic data seems to say it should not.
You're correct. But who are you to believe? Science or your own lying eyes?
I think the kinetic data is more reliable than this other stuff.
Well, see, we have the same problem with the little critters, the salt bacteria that are found in the Permian of New Mexico, where they said, but we cleaned it all out. We used acid, we used alkali, we killed all the germs. It can't possibly be contamination from outside. So we have to be able to get 250 million year old bacteria to resurrect. And the other side is going, but it can't last that long. There's too much radioactivity, there's too much this, there's too much that. It should have decayed by now. And it's the same argument. See, and this is the thing that we run into this again and again and again. Mitochondrial leave. Well, if you believe the modern rate, mitochondrial leave happened at 6,000 years ago. Funny they should pick that date. But if you believe that chimpanzees and humans derived from a common ancestor, in other words, if you use an evolutionary time scale, then mitochondrial leave was actually more like 200,000 years ago. So, do you believe that the present is the key to the past, or don't you? If the present is the key to the past, then mitochondrial leave existed at 6,000 years ago.
There's significant geologic evidence that the past is the key to the past.
Yes, we have another.
Life exists without it. I mean, it's in all the cells. It needs the energy. So, if they say something is older than even that, how does that make sense? So they're limited to that just by their own logic. Now it's getting shortened down even more. These people are really in trouble.
Well, that's right. If you assume that modern processes happen at the same rate now as they did in the past, then I think you're starting to get some arguments that the past isn't that long ago. On the other hand, if you assume that the past had to be that long ago, then what you're going to come up with is the idea that rates in the past were slower than they are now. In fact, we've heard that in amino acid dating too. The further back you go, the slower the decay or the racemization constant for checking things out is. You know, when I'm starting to see this pattern, I'm starting to see people who believe in uniformitarianism, their uniformitarianism is coming around to bite them. Yes, comment here.
I think it's interesting that they could smell what was in those bones, because those are volatile organic compounds. I would guess that they're pretty labile.
And the question is, why aren't they all diffused out by now?
Yeah.
It is an interesting question. That's just another part of the same problem that we're having. And of course, for what it's worth, some of these same bones were carbon dated, and the carbon dates are consistent with the creationist scenario and not really consistent with an evolutionary scenario. So we're starting to see a lot of things that are pointing in the direction of short age. That if you're really going to be fair, you got to say it's probably not that old.
It's great.
Well, anyway, next week, come back. We will see what happens, but hopefully we will be into pseudofossils, although with a lot of things that are going on, we may take a detour before that, but we will see.